नहीं ऑन करना
ऑन
कभी हो गया बोले सर आप लंच पे ऑन कर can just go someone sit there you can see all of them right no, wait. This is fine make a circle
Yeah, is it on?
We just turn it off.
Just switched off again.
No, it's on. Now it's on. It's fine. It's all of them.
Welcome you all for the next session of second day of molecular biology meeting. So now we have a panel discussion which will be chaired by Professor Samitra Das, and the panelists include Virology and Sunita Bhattacharya from TISTI and Rakesh Mishra from TIX Bangalore and sorry.
We'll have to next year.
No, I think sir.
Also.
Welcome back. I think some people are still relishing the dessert. Thanks for sacrificing the dessert. So, as I mentioned that in the opening remarks, the genesis of the molecular virology group, and I'm very glad that I could get at least one of them here is Professor Virology, who is the pioneer in the country along with Professor M.S. Shaila. And they used to have collaborations, and then he used to frequently meet with IIC and work with Professor Shaila's group. And that's the time during the centenary we thought like we'll have a virology meeting, and that was the genesis. Anyway, I won't repeat. So I'm very glad. Thank you, Dhruvoda, we call it, with full of respect. Professor Virology Chatterjee is now currently the Vice Chancellor of the Sister Nivedita University in Kolkata. Before that, he was Vice Chancellor of MET Kolkata, and prior to that, he was OVC for Calcutta University for many years. And we are all trained in his hands, and there are several virologists sitting here are directly or indirectly benefited from him. We have in the panel Dr. Sudhanshu Broti. Sudhanshu Broti again doesn't need any introduction. He was actually, until a few months back, he was the executive director of the THS. Initially, he was in THSTI, and he was the executive director of RCB, Regional Center of Biotechnology. We have Dr. Sudhir Krishna from NCBS. Again, when I talk about the virology, the first person I met in Bangalore and the first visit was actually because of the invitation of Sudhir Krishna. So I think it's a good reunion for all of us. We have Rakesh Mishra. Rakesh Mishra, many of you know as he's an expert in epigenetics, but today he will show that other facets and how he has evolved over the time and in the disaster response for the COVID. We have Jayant Bhattacharya. He is now the director in charge in THSTI, and he is again another very, very renowned person in the area of HIV biology and HIV research. And of course, so what I thought that just to structure this panel discussion, although I want all of you are actually member of the panel and then you will feel free to interact. In the beginning, I will probably invite, first I invite all the panelists, please come and then take your chairs. Dhruvoda, Sudhanshu, Sudhir, Rakesh and Jayantu. So I thought that in the beginning I will just invite all the panelists to have 3 minutes opening remarks, and I have allocated some topics for them. Dhruvoda will actually highlight the molecular virology research for the past 30 years. Past 30 years in India, and then Sudhanshu will talk about the virology education and the challenges of having a virology program by itself, a BSc level or the MSc level, that she's going to discuss. Sudhir will discuss about how to train for the future virologists. Many of you are actually mentioned that you are actually a student participant in the first virology group meeting. So that percentage may be 25%. We have to make it almost near to 100% of these people to take virology as their professional career. And Sudhir is going to highlight what he feels about the training the future generation virologists. And Rakesh is going to talk about, we have the commitment for the society and the societal needs and the community. So how heading the TICS after heading CCMB, he has now moved as the director of TICS, which is in the campus of NCBS. He's going to highlight how his institute and his own perception towards the virus research, keeping in mind the community and the service to the society. Jayanto is heading translational health science research institute THSTI. So definitely he is the right person to talk about the translational scope for the virologist. He is going to do that. And finally, I am going to talk about how networking, my experience to head the INSACOG for a long time, and now actually advising the INSACOG, the advisory board. So I was just trying to see how I can share my experience for the benefit that I got from the networking, particularly national and the international level. And after the opening remarks, we will elaborate in some of these discussions. And finally, I will leave it open to the questions and other issues that needs attention. So I just clustered like first 15 minutes we are going to talk the opening remarks. Next 15 minutes is the elaborate on those subjects if they want to have add on something. And the last half an hour I kept it for an open interactive discussions. You are allowed to ask questions de novo or any of these things that I mentioned. If you have any suggestions or you want to have questions any targeted to any panelists, please feel free to do that. But as I mentioned in the first hour, that this is a group meeting. Don't take it something like a very formal conference. Feel free. We have very little questions from the students. I want at least in the last hour you just try to have some questions. Okay? Let me see what is in your mind. With that, I would invite, it's my real pleasure to invite the pioneer of the molecular virology, Professor Virology. Thank you, sir.
Thank you, Shobhitra. Now, as mentioned by Shobhitra, that to give 30 years of molecular biology in 3 minutes is something which I think is a little bit difficult. It's a bit difficult. So I'm not going to tell you the history of that. But I can share with you some of my experience because I am, after coming back from the United States, with training in the animal viruses, rhabdoviruses. When I joined in 1989 at Harvard University. So at that particular period of time in our university, we don't have in cell culture, tissue culture lab also, but I joined and I thought I should work with animal viruses. And naturally what I have to do, I have to try to find out who are the people working in this area who are working with animal viruses. And I actually contacted number of institutions at that time, but most of them said that they are more interested to do, which is another very important aspect, but it's an epidemiology of viruses. They're not interested in the molecular virology of them. Except the Indian Institute of Science, at that time actually I am defending one of my projects, and then Professor P. Padmanabhan was actually the chairman of that particular committee, and he mentioned to me, why don't you contact the Indian Institute of Science Microbiology Department and discuss with them whether it will be possible for you. So that's the story of myself to get in touch with Professor Shaila. I'm missing him today. I thought that he's going to be here. But Professor Shaila...
Next is Professor Shahid Jamil.
Yeah, no, I'm coming to that. So Professor Shaila then actually tremendously helped me to actually set up and discuss and guide me in different aspects of this, starting my career in the field of molecular biology in India. Now then, when I'm working and setting up my lab, I'm doing a little bit of work and other things. In 2000, I think 2002, no, it is in 1998. That is almost 10 years of work has been trying to work and others. In the meantime, ICGB, Professor Said Jamil, and there are another group who are working in this particular area. And the reason why I'm mentioning Said Jamil is this, that there is a conference at that time going on, which is called the Buha Research Conference, and in that conference he also learned, myself also presented our work, and thereby we have started interacting, and in fact Shahid Jamil helped 2 of my students to work in his lab to actually develop this particular. So you can easily understand at that time molecular virology is really not a very well discussed things amongst the virologists who are present at that time in India. I should also mention the help I received from National Institute of Virology in the respect that I just looking at a virus which is very India specific, with which I can work with, and you know that by going the literature, I could find that there is one virus identified by, at that time it's not NIV, it's NICMR Institute. So, there is a virus identified from Nagpur, the Cheripura virus, and then I have requested the director of the NIV, and he actually immediately helped me and said, I am going to give you the strands, I am going to give you the cell lines also where you can grow these things. So that's something which I should mention that helped me quite a bit. But again, he mentioned that in my organization also we more or less stressed on the identification and the other things and the distribution of these viruses. After that, in 2000, as I am mentioning you, another 5 years after that 2000, there are a number of conferences where we used to meet with different people at different places who are, in fact, Sudhanshu, if you remember, in 2002 actually we met in the first conference where he's actually working at that time with JEV, and he mentioned, and also in stroke science there are a number of people who are working in that area, and we started actually discussing about what could be done, what are the different things which we could do to strengthen this particular molecular virologists who are almost very small in number. And then we thought about in 2009 the first molecular virology group meeting has been done. So first molecular, we are right now in the 7th molecular virology, but the first one has been done in 2009 organized by the Indian Institute of Science. And at that time, the number of institutions from where we could get the participants, I remember it is around 30 institutions out there. And Dr. Rani is here. He is mentioning about the number of organizations today participated and presenting the paper. It's not 25, it's 47. So if you look at it, the number is gone, we're doing. And also I came to know from Shomitra that there are large number of papers which could not be accepted at all. So at that time we thought every 2 years we are going to do the conference, so every 2 years we have done that till 2019, and then for COVID we couldn't do it for the last 4 years, but in the meantime you can see the huge amount of research in different parts of the country with the help of the stalwarts who are present over here and also who are not here, help the molecular virology to grow in a very big way. And now specifically the demand is coming, why every 2 years, why not every year there will be opportunity for the people to present their work. So that shows that the importance of the molecular biology right now understood by the Indian community in a very big way. And I'm sure that during further discussion I will come back because I have already spent my 3 minutes. Thank you.
I'd like to invite Sudhanshu to highlight on the virology education.
From here or there?
So, opening remarks from here, then we can discuss.
Including the
So, good afternoon. I've been asked to comment on possibility and feasibility of a virology structured program, academic program, considering the importance of virology now. Actually, virology was not a sought after area in this country until COVID happened. And COVID turned a lot of plant scientists also into animal virologists, so that was great. And that is why you see 47 institutions participating. So it is good growth for molecular virologists in this country. So I come from Regional Center for Biotechnology. RCB is actually a university through Act of Parliament. That means that we have the authority to grant degrees and impart education and training courses. RCB has been running master's and PhD degree programs in biotechnology as such. So our board of studies, were also fired up that we should start a virology program here. And we did consider starting a master's program in virology. As of now, I think there are 2 programs of master's degree in virology. One is offered by Shri Venkateshwara University and another at NIV. NIV does it in parts. No,
Yeah.
So these are the 2 virology programs. But we worked on this suggestion from our board, and we figured out that in this country, unfortunately, when you are looking for a job in a university recognized by UGC or Government of India, there are prescribed qualification. Like if you are going to Delhi University, going to Department of Zoology, if you have a degree in biotechnology, you are your person not wanted. So they need a MSc in zoology, or a Department of Botany will need a MSc in botany. You can have PhD in anything, perhaps that is tolerable, but MSc, the primary degree has to be in that discipline. So we figured out that if somebody gets a MSc in virology, they will be jobless. That's it, there are very few jobs, and then we thought, these guys will come back. And I remembered one such incident. DBT insisted that we start a course in translational sciences. And an MPhil program was started at King George Medical University in Lucknow. And the first batch, I was asked to go and meet them after they had completed the year. And they were asking, where do we get the job? And I had so much difficulty telling them what to do. I escaped from there. So I realized that starting a MSc program in virology is not something which is desirable. But yes, what we did in our biotechnology program, and I have made this recommendation to DBT for other biotechnology programs, that there should be a specialized course on virology, including molecular virology. We have started a 2-credit course. Of course, there used to be a little bit of virology and microbiology, but we have kept microbiology, everything except viruses, and we have made a specialized course at our institution, molecular virology. Actually, the course name is molecular virology. It talks about viruses, virus replication, making viral vaccines, antivirals, all that molecular stuff. So, as of now, my recommendation, my suggestion is that let's not worry about starting a master's degree program in virology, but we should restructure our biotechnology or, there is an MSc program in molecular biology, I don't know where they get the jobs, but we should start a virology course in all programs, that is zoology, master's degree course, they should have a virology course. Even botany, why not? Plant viruses are equally important. So there should be specialized courses, and this recommendation actually we are also making to DBT to implement for the program that they are supporting. So I'll stop over here and I'll take any questions. Thank you.
Now I have the pleasure to invite Dr. Sudhir Krishna. Sudhir has actually already shared his thoughts. His 2 questions. Number 1 is, how do you train and support a virology career in lab and in community? The second is, how do you balance team, individual science and service and education? Sudhir.
Thanks, Amita. I was talking to Guru over lunch, and I made a sort of slightly facetious story, but I was saying that, when I was young, we didn't have IPL and one-day matches and all. We had only test cricket. And Farooq Engineer batted the whole evening and delayed the match for the next day. And he was asked in an interview, and the details are not important, the concept is, and why did you delay the match for the next day? And he said, I had 50 rupees. He did it for 50 rupees. Today, Virat Kohli, if you call him to give a talk, he can buy the neighborhood and not even notice in his bank account. I mean, the point that I want to make is that I think there is a crisis in science. I don't think that the crisis is of science. I mean, if you look at individual talks out here, and I've been hearing it for the last 2 days, Raghavan had organized a meeting. You can sit in any global forum, and I haven't been to meetings for a long time, and pretty much you'll hear the same talks. But Guru was saying that you're struggling to get funding for this meeting. So there is, and you are very influential people like Rakesh out here, so you should ask people like Rakesh, what is the connection? I mean, very influential administrators who know this. The question that you should be asking him is, what is this disconnect that we're seeing right now, where the level of science, where the quality of individual presentations is on par with anything else, but you operate at a sub-threshold level. And in this August audience with Tata's photograph there and here and there, I think this is the right place to be asking that question. So I know I did send you those 2 questions, but the question that I will ask the panel and ask is, why is there such a disconnect? Are we caught up in insularity? Are we caught up in process? Are we micromanaging our faculty all the time? Do we check whether they go to the bathroom 3 times a day. I mean, what is it that we do which reduces us to such a sub-threshold, and everybody does it in spite of it. You ask anybody to give a talk out here, they'll say, in spite of everything, we got this done. I mean, they don't tell you that because of this, we've got it done. So I do think I'm exaggerating, maybe a little bit, but not too much. I mean, if I'm exaggerating, it's to make a point. And so I hope the panel and, as I said, influential people like Rakesh will take this up and sort of address some of these questions.
Your original question.
My original question will come to in the discussion. I don't want to take up more than 3 minutes. So next I would like to call upon Jayanto to share his views on the scope about translation of research. That's my job.
It's a privilege to share this stage with all the dignitaries, especially Dr. Virology, who used to be my teacher, and who actually I learned the RNA virus and DNA viruses. I still remember that class in the Bose Institute, where he actually helped us learn how do you distinguish fluorescence RNA virus vis-a-vis minus RNA virus. I mean, actually that fascinated us a lot. And I still remember one of my classmates asked me a question. If there's fluorescence RNA virus, you have a RNA polymer that goes in 1 to 3' polymerase and this classification. How about the minus RNA? You don't have. And he started explaining how the host comes in and play a role in giving a polymerase that can be useful. And that's the start. So that's so when you realize that any time sharing is great. Thank you for the opportunity. What I'm trying to tell, I just want to tell you a few things. I just don't think I have anything to say, anything more than all of them to say. I want to give you my perspective of what I learned in the last 20 years working in the area of, primarily in the area of viruses and to some extent bacteria. After my postdoc, I did my postdoc in the area of HIV and before that after viruses. And I joined the National Research Institute in Pune in ICMR. 7 years I spent there. I decided that's all. It's enough. I just don't want to work in government institutions anymore. So I decided to move to sort of industry. It's not an industry. It's not for profit organization. But it's like it was in the principle of industry as IOI. That was actually prompted by none other than Dr. Bhan, who was our section. And that led me come to DHS in 2013, so I got past the years here. And then I worked in this DHS with my employer as I have, but I enjoyed being at an academic institution. So on one hand, the pressure of working in the corporate pressures from the headquarters in New York. We have 4 labs, and it has actually 4 labs, one in India, 1 in US, 1 in London, and a lot of clinical research centers. And then very recently I decided that enough industry has come back to the academy. That's what I did. But what I learned, there are 3 things. One is we create grassroots study wisdom, which means that if we're talking about translational research, it has to be bi-directional in nature. You can't just go and, I don't buy this argument that clinicians are not right person to go on. It's just that we don't know how to communicate with clinicians. That's the high point. And if we know how to talk with clinicians and to conduct the clinical trials, then we can do what HST has shown in time forward. That's all I want to say. Why I'm saying this? Because I still work a lot, not directly but indirectly to industry. You know the industry, what is the mantra? When you heard about, to get things done in 100 days. If it is 100 days, the first 50 days, they will ensure that they learn how to fail. You need to learn to fail, because unless you fail, you will never learn. So those for the young people here, when you take up a project, you should accept your failure. You must fail. If you don't fail, you will never learn. It's not easy to make an environment vaccine just because it has worked out, right? I have a scientist, a colleague from the HSJ who actually told me how to modify the environment vaccine by making the circular RNA. So there are a lot of, and that all happened in accident, right? So that's what I wanted to convey, that if you want to do a translational research, it has to be bidirectional in nature. You know how to communicate, you need to build capacity, and you need to work in tandem. And then only you can expect. I would stop. Thanks. Okay. No, that's why I just had a gap in between.
Thank you so much for inviting me. Again, yes, thank you for that. So, my job is to talk to you about what society expects and how we can help society and connect. And that is partly addressed in Sudhir's answer, that if we connect society, some of the problems will be less painful funding. And we have seen during the pandemic how our science has really helped in a real-time way to society, and we are still continuing to do that. As Mr. mentioned, INSACOG is still active, and to many other consortia or other methods, still surveillance of the COVID is going on. And particularly this city, we have been doing, from our campus every week viral estimate of all 28 STPs of Bangalore, and we have been so cooperative, and we tell them every week what is the load in which STP means which ward, and we give them the warning of the fourth wave which none of you noticed because it was not symptomatic at all and we were all protected. But we could tell the PVMP before why the wave will come, and that has created a good relationship with them. That it is the only city that puts data even in the internet. This is the peak and this is the wave and so on. So that is that science has helped something that we learned. We could tell which variant it is. Suddenly a peak is coming, so we know the variant is not new, then why peak is coming. Then it turns out there was a Sahara, there was elections ID or something. So you can relate to social practices and our events to what is happening at the disease level from the environmental sample without touching anybody. So that is very, very useful thing. And but if there is a new variant, then now we have to have different strategy because it's going to create a problem. So the connect of society to municipal corporation and to people is so it was so well established during this time. And we tried continuing the same thing for dengue, and we never got consistent results from the wastewater for the dengue. Now, so we go back to science and we see how virus is shed for the COVID and how the dengue virus sheds, it comes only through urine. SARS-CoV-2 has to go to air, it has to be so much in quantity that people breathe and it spreads. While dengue has hired a taxi of the mosquitoes, they come and so the quantity of the virus is so low that you will not detect in water. So we change this strategy, we now go and collect mosquitoes and larvae because this actually it can be transmitted to the larvae from the female, and we start estimating in which locality how many percent of viruses, a mosquito are carrying the dengue virus, and relate that with the clinical data and try to establish, can we just by doing vector surveillance tell how many people in the city are having the infection. Because dengue, again, various kinds of symptoms, most of the people don't go and report. So actually what you see the report is maybe 10% of the total number of cases. But if we develop methods, and similarly now there is a lot of emphasis to monitor these viruses from the zoonotic sources, from the cattle, from the poultry farms, and maybe where the human wildlife conflict zones are there. And we can keep a track of what kind of pathogens are in which reservoir, and if they are spilling out, and if they are spreading into people, how early we can, if we keep alert system. I mean, there is a lot of debate. If China told about the world a little bit early, maybe the trajectory of this pandemic would have been very, very different. So if we are aware and we know and take immediate steps, then we are really directly connecting what is happening in the lab, the highest quality science, mutations, variants and so on, and at the same time how it can translate into strategies of controlling infection and taking care of the public health. So that's something very, very, I mean, we have gone through a lot of trouble and pain. All of us must be known people who died during COVID. But the trust of society that we have gained during this process in science is enormous. And I think if we capitalize on that, both at the decision-making level and at the societal level, it will be at least one good outcome of the pain that we have gone through the past 3 years. So I will stop there and then we can discuss more. Thank you.
Right. So, as I mentioned in the beginning also, what we have learned from this INSA talk during this COVID management, that how India can achieve things which is almost like unachievable just by collective and collaborative research on nationally supported projects, establish international collaboration research strategies, and in an interdisciplinary perspective. And as I told you that it's not only the genomic surveillance, also in the vaccine development, also in the trying to have a disease management. Every sector we have shown that how important it is to identify the expertise of a cluster. You identify the strength that you have, and then you try to identify the strength of others and collaborate and get the things done in a stipulated time frame. Otherwise, it's very difficult to have an individually achieve any goal. So I think that we need to realize. One more thing I just would like to highlight, which you can discuss because it's the right forum. Recently, we were in a meeting, the India-US strategic dialogue in White House, and this was regarding about the biosecurity, biodefense, bioethics. The main problem of the basic researcher is where to draw the Lakshman Rekha. I think one of the scientists from Stanford, he mentioned this word, Lakshman Rekha. Being in Stanford, he agrees that there is something we need to learn, where you need to stop and where you need to do it in BSL-3 or in BSL-4, where you need to report, as Rakesh was saying, if there is any accidental leakage, timely manner it should be reported, docketed, so that people can be alerted immediately. This is not only a national responsibility, it's an international responsibility, and it's a humanitarian responsibility. You just can't damage for your own callousness. So something that also we need to take it seriously. I think what we'll do is we'll go one by one issues and we'll just ask them to elaborate a little more, and you can raise your hands if you want to have any questions on that aspects. First thing I will start because Sudhanshu has to leave, his slide is going otherwise. So on the virology education, Sudhanshu, you want to elaborate more or you want to take questions on what people feel about how difficult will it be, because many of you are actually handling in the different research institutes and in universities. How difficult will it be to implement that virology course is mandated to learn? Even if you are doing MSc in botany, MSc in zoology, there should be some kind of a minimum requirement to have a knowledge about. This is an important component of the infectious disease, and that way if you can make it. So,
no, I don't have anything more to contribute to this, but I'll be happy to take opinion and perspective from others, how they feel about it. I have told you the challenge in terms of employability in the Government of India universities with the MSc Virology degree, but I am all for virology. But if somebody has a solution, we'll be happy to discuss.
Let me tell you, of course, I am not trying to promote introducing virology. I am an engineer. I am an alumni of this institute of PhD in aerospace engineering. I am a US citizen. And in fact, we discussed several years ago that I was at Wright State University as a research professor and in the mechanical engineering department, mechanical materials engineering department. There are Wright State University and in fact Dayton area, they have hospitals, many hospitals, and Wright State University has a medical school. And how they do research is, in fact, they started the School of Interdisciplinary School of Science and Engineering. I think President Obama, he I think granted about $100 million he funded for doing research on neuroscience and engineering. And Wright State University also got a big funding and they started a neuroscience engineering department. And they have a medical school, many of you may know. And the way they do research is they don't have a big hospital in Wright State University. They collaborate with all the hospitals there. And that is where I see some gentleman was talking about industry-university collaboration. And that is lacking in India. In fact, I pointed out here to Professor Balram a few years ago. And see, there is no biomedical engineering department in
We have it
Institute of
now.
Science.
We have a bioengineering department.
No biosystems and science. There is no biomedical
Now we
engineering.
changed the name to bioengineering.
Bioengineering, yes. Okay, it's all right. In fact, I met, we are talking about Professor.
I think Naren is here, chairman of that department.
Huh?
I'm the PhD student.
Okay, I met Professor.
I think you come to the point, the virology course.
Yeah, let me tell you, the important thing as an engineer, I would say, while whether it's virology or whatever field of medical science, you are related to medical science. And you should have a course in engineering analysis, because collaboration with engineering is very important. That is very important. I see that is not there in the US universities also. In fact, we suggested discuss about it with the State University.
So you have any idea about both coursework? Right now we're discussing about virology as a
Yeah,
discipline.
even virology or medical sciences, they should have an engineering analysis, because you should find out how to analyze the problem.
I think you can
stop
just
there. I don't
continue.
take up much of your
Thank you.
Thank you, Professor Murthy, and I think very nice suggestion given by you. So this can be done at two levels. One, introducing virology in the MSc Biotechnology program. There are few universities, entrance examination is conducted by at central level, probably CT is there as a controlling body. So they can communicate to those universities or they can call a meeting where such kind of changes can be made. And there are very many universities which run their own course of MSc Biotechnology. So I think that can also be, this message can be passed on through either through RCB or DBT, that they should to have a kind of harmonized course curriculum they should include it. And you are very right, you won't believe that I designed a program for MSc Virology in Banaras University. I got it approved by academic council, but due to same reasons I never implemented it. So this is very much true. Yeah, thank you very much.
Any other comments on the courses on Institute of Virology? You have something to do. Anthony, you wanted to say something?
Thank you, Sumitra. I am a retired person, but I still have so much of love to virology. And
You taught
I
virology
was-
course for 20 years.
And something is that Sumitra, Saila and myself started the molecular biology course in the institute because the graduate students who comes to IIC has no idea about the viruses. They did not know how the virus replicate, what are the types of virus we have, and three of us discussed this matter. I'm not a full virologist, Sumitra may be a full virologist. And we decided to have the molecular biology course. And Okay, so I was under the impression that this panel meeting will tell us the direction the government agencies or government institutions should take maybe the next 10 years or 5 years. So that is what I expected, that is what we should discuss. Because I don't think any of the things which you discussed will impress the young scientists. What they want is what direction we can give for us to do things for our government. So I think I appreciate Sudhir, who told us what we should discuss.
Right, so we are now coming to Sudhir. Thank you. Sudhir, because
Maybe
we
I
have
can,
short time,
if
I
I
will cut
can,
you short.
Yeah, but
Anything else?
time is
I know.
important.
Exactly.
But we should have some sort of a structure and guidance what to do.
Right.
So
Maybe now since the government is
Thank
making
you.
any courses can be attended by any people, you can do, there are lots of heterogeneity that is coming. So we should maybe work that UGC removes this crazy thing that for biochemistry professor you need biochemistry master. That is much simpler than trying to follow the expired material and try to adjust with that.
Right. So thank you. So a couple of things which I think would really help. One is inter-campus efforts and removing this. I think there's an obsession with campuses. And I think opening up electives, whether it's engineering, whether it's across, and having inter-campus degrees significantly. I had the privilege of giving a talk in a school where they said that I gave the shortest talk as a chief guest, and I said only one thing, and that was this idea that we are one thing is just not true, and this whole education system teaches us that we are one thing, and it's really not true. And I think places like the University of Science, with your influence and so on, should really start reaching out to the fact that we are many things. I mean, we are not doctors and epidemiologists or molecular biologists. We're a bit of everything. And specialization in the last 25 or 30 years has come at the cost of perspective. And you saw that hugely in COVID. And one of the ways that you can deal with this whole issue of the loss of perspective is actually by immersions, off-campus immersions, and so on. So I would strongly encourage a loosening up of the system locally, so that you think of leadership as a growth process where people have early networks, early immersion experiences, and so on and so forth as it were. But one sort of bigger point that I would like to make is I really think that we should go global. And this idea that we spend all our time being subjugated of committees which has flown in from US and jet lagged and telling you that you're this or that, take on global challenges. I mean, as I mentioned in my introductory remarks, the quality of the talks here could match anywhere. So what is stopping us from taking on global challenges elsewhere and just diversifying? I don't understand what's stopping us. What's stopping us?
What I just wanted to mention that, yes, I completely agree with Professor Krishnan. The reason is, so for example, in our Calcutta University, as Shobhitra mentioned, that there are so many of my students over here who are right now virologists. And Calcutta University, we could introduce MSc Biochemistry as well as Virology. And that's a component, and that's there for last 30 years. And now, definitely what was mentioned here, that recent initiative in the NEP 2020, it has given all the scope to get it incorporate whatever cross-disciplinary subjects you want to, you can get it, and it could be done, and there are all possibilities are there. There will be no problem. So the student of even the students of social sciences, they can take the courses in computer science also. So all these possibilities are there. So now we the faculty members, we the teachers, we have to actually incorporate that. And unless we actually make this effort from our part, it's very difficult. We have to actually introduce these things, and we have to take this opportunity of NEP 2020, which will definitely help to get these cross-disciplinary things. And you'll be very happy to know that in our university, the number of engineering students opting for dance as their elective is highest. So there are possibilities out there, because that is what actually we have seen it. So there is no restriction for these things. These things could be implemented. Thank you.
In fact, let me tell you, my field is turbulence in aerospace engineering. I'm a mechanical and aerospace engineer. And I play flute without flute and experiment in turbulence. So you mentioned about dance. I play flute without flute. Started practicing since about 10, 12 years. So, I mean, I don't know I just try to simulate the spectrum, the turbulence spectrum or whatever. I only can accompany good musicians, classical or any other music. But I do it as an experiment in turbulence. So there is this day and age, there is no boundary between one subject and another. Cross-disciplinary and interaction with universities and research establishments. That is also lacking here, that's my feeling.
But basically, I think all the majority of the head of the institutes are here. You have to, I think what Sudhir also said, that you dissolve the boundary, identify the cluster of expertise, and then reach out and get things done. And there's no discipline as such, interdisciplinary or cross-disciplinary, that's going to help you to achieve the success. Rahul?
Yeah, I just wanted to just highlight the same point that we are discussing whether modern...
Rahul is in chemical engineering. You hardly stop today.
No, I just wanted to say that we should not be thinking about whether we should teach molecular biology or molecular virology, which is even more niche to people or not. I think anybody should be able to learn, including me. This process, we should be thinking about how to make the modern leaders for the next generation of problems that are going to be real, and this is what the leader is going to point out. And we should be addressing problems which are of societal need, because then we automatically will get supported by government. So we can convince government, we can convince decision makers, we can convince the public that what we are doing is really going to impact their lives. Then we should be able to gain whatever funding we do. I don't see a reason why we shouldn't be able to do that. And of course, we should go global. There are tons of challenges, many problems which are outside the country, which are equally important for the rest of the world, which I would say we should not be kind of shying away, because the Indian government is not going to fund those things. I'm sure there will be funders outside who will be able to do that.
So, thank you, Rahul. We were talking about translational research, and I think before getting into that, many of you know that we are having a small epidemic in Kerala with the Nipah virus. I would like to hear from Sri Kumar what is the situation, and then how you are handling it, and then as an he is heading the Institute of Advanced Virology in Trivandrum. So I think he will, from the field, so he can apprise us what is going on there.
Thank you. Actually, the Nipah virus outbreak in Kerala is under control. So it's absolutely under control. There were only, this time we fortunately had only 6 cases. 2 people lost their life, and 4 people could be treated and brought back to life. Actually, there was one kid of 9 years, and he was in ventilator, and maybe the clinicians were, at least they have learned how to manage that. In the 2018 outbreak, there were around 21 cases, 19 people died, and only 2 people could be saved. But this time the trend has reversed, and they have found that instead of using ribavirin as a treatment, this time they used remdesivir, and it is showing some positive response. But one thing what we could see is there is a change in the clinical pattern. In the 2018, it was primarily an encephalitic, a cluster of adult encephalitic cases that happened, and this time it has become predominantly a respiratory kind of infection. So the chances of spread was much more this time, but maybe we were aware of the condition much early. And again, the same clinician who diagnosed the 2018 diagnosed this case also. That means there is a lot of clinical acumen or intuition is required to pinpoint, because already the monsoon is going on in Kerala and there are large number of respiratory cases. From that, picking up one particular case that is also showing a respiratory symptom predominantly as a Nipah, or even suspecting that as Nipah is a fabulous clinical training for that particular physician. So now coming back to recently, after the 2017 Zika virus outbreaks in Kerala, the government of Kerala wanted to have a local expertise or an institution which can focus on virology itself. And they started discussing on this. In 2018, they established a new center, initially with a 30,000 square feet building and now with an 80,000 added to it. It has become a big facility in itself, but it's picking up. So 2021 onwards, the institute has started functioning. There are 8 departments. That's the Institute of Advanced Virology, exclusively dedicated for studying viral pathogens. The mandate is to study anything and everything about virology. And we can study any kind of viruses, including plant, human, animal, marine, fisheries, or whatever kind of viruses. But the current focus is on medical virology, and definitely the government of Kerala wants something that is to be translated. So I would like to connect something with what Dr. Sudhir was telling. So of late what we are seeing is we are losing the perspectives, and if you ask any of the students who is sitting here, I'm sure that 90% of them will say that they didn't want to study virology, but they happened to come to the virology because they didn't get cancer virology. And maybe after the COVID, the trend would be changing. Because now people start feeling that okay, virology is also a subject which we need to invest much more, and also of late we are seeing that there are solutions coming in. See, most of us see all this as research problems, and we never project it as a research solution. So now we see that okay, we can make vaccines, we can make antivirals, and we can control or identify, control, and treat these viral infections. So there is an added interest. And I'm happy that the government of Kerala has taken this initiative, and maybe other states also will follow suit, so that we can, instead of focusing on the molecule, on one molecule. See, previously maybe the first generation virologists who were trained by the classical virology, maybe people who are trained under classical virology, the first generation virologists of India, they used to culture the viruses and start from there. Or maybe they used to go to the field and identify a patient or an animal who is infected, take the samples, bring it to the lab and isolate the virus, culture it, and learn or do all the classical techniques and understand or feel the virus in their hands. I can vouch that maybe 50% of the new generation virologists have not cultured a virus. They would have taken a virus.
RT-PCR was not common at the time. So I will have to cut it short.
Okay, I'll just stop with one more point. This is one thing that we don't learn the classical virology techniques to feel the virology and to sense the virology. Second thing is, those people who are doing advanced virology, they find it very difficult to translate this into the field. So that is another point that we need to take into. Like, I have a molecule, or some of you have a vaccine candidate, so how to translate it into the human population? We don't. It is not collaboration. Within the country, how will you do it? How much of clinical trials we are doing? Or how much of us are equipped for doing clinical trials? Maybe who have done this, the translation kind of virology would know this. So these are some of the inherent problems that we are facing. But definitely there is a big challenge ahead of us that also leads to big scope for all the new students.
So clearly we cannot solve the virus research problem in 45 minutes. So what we need to meet again. So we started the journey in 2009 at ISC, 2011 at NIV Pune, 2013 in RGCB Kerala, 2015 in ISC again, 2017 THSTI Hyderabad, 2019 IIT Kharagpur and Kolkata institutes along with them. 2021 we couldn't meet, 23 we are meeting here, and we are requesting Dr. Sri Kumar, none other else, because he is heading an Virology Institute, to host, and he has agreed because I don't think he needs permission. So now the question is, this year, because of this time and other constraint, we couldn't accommodate 50% of the abstracts. I am apologetic from the beginning, and so is Shashank and
Keshav, we wish that all of you, who have shown interest and now watching, could have been accommodated. So for that, next year we are just trying to repeat this meeting, and I would vouch for the people who could not have the platform presentation given preference, but there will be a mixture of both, so that we can accommodate more people, and after that we take a call whether we should go back in alternate year mode or not. So just because we could not finish all of them together and time constraint, we can have next year maybe in the mid-September or third week of September, so that we can lock the dates, and Sri Kumar, if you agree, then we finalize that on that meeting. Thank you.
Then I would be very happy to agree to it. And since this ours is a new institute, and I would invite all of you to come and join the next virology meeting. The last virology meeting in Trivandrum was in 2016 at RGCB. We could do it well, and the feedback was extremely good. I hope we can match to that level. But being a state government institute within our own constraint, but we will try to do it in the best way as possible. And I will be very happy that you come over and join, and we'll have a wonderful session there. And I would also like to have one session on where this industry can participate. This time we had only one talk, but maybe we should have more participation from the industry.
Essentially we need 2 full day or 2.5 days. Looks like now the family has grown bigger. Nothing to do with the COVID, because those I say overnight biologists. Overnight biologists were not that many here. But the core virologist also has increased, and that's going to increase again. So I think we'll leave it at that. I think we are running late, so and we are having tremendous pressure to go to the next session. And I think
There's a question there.
Question?
Yeah. Actually, since we are all scientists, not me, but there are so many scientists sitting here, I'll speak on behalf of most of the PhDs and postdocs. We have this constant conundrum, like whether we should be in academia or we should join the industry. And considering the funding situation in India and the age limit that we have, to be able to apply, I really want to hear what you all have to say about this.
Can I?
For sure. Okay, anyway, we will share the next session. Yes. You will cut into your time now.
Alright. Good question. So, because of COVID-19, thankfully, there has been a huge virology education. Now the onus is on the next generation students to not let it die down. And industry, as an example, Ajay is here, you have seen the talk, the kind of impactful work can be done. Fantastic opportunity to open your everything beyond academic
Decide the next
absolutely, yes, it's a project. And not looking abroad, looking inwards also. And to go it again, we have a lot of capacities, a lot of companies have developed virology wings, which were not there before. So opportunities have increased. It is for you to cash it out now. Onus is on you. And please do a good job in carrying it forward. So we'll move now.
Before we move on to the next session, I would like to thank the panelists and the chair, Professor Das, and all the panelists for such an enlightening session. Now let's move on to the next session on short talk. Thank you. Will it stop or should I stop after some time?
Alright, so this is the last session with the talks. These are short talks, and the speakers include faculties. Their talks we have to squeeze in short format. Apologies for that, because the program was too tight. And then there are a few student talks as well, which were ranked highest based on the abstract scoring, not on the poster scoring. That's different. That will be announced right after this session. There are 5 best poster awardees that will be announced. But to be on time, I'll introduce the first speaker,
Move that.
which is Dr. Vainav Patel, and he comes from National Institute of Research in Reproductive and Child Health now, and he did his PhD from University of Bombay, then training in NIH. He has dabbled in both DNA and RNA viruses. Today, let's see.
Yeah, that's
he's going
better.
to talk
Set the
about
timer
CMV.
and put it on. That. 12.
Short talks are for 12 minutes, ideally 8 minutes for presentation and at least 4 minutes for few Q&As. Professor Vainav.
Timer is better. Okay, so are we? So quickly, I'll get the thank yous out of the way. Most grateful to Jayanta, Professor Das, Keshav and Shashank, I guess in that order, the squeezing or whatever happened, I heard. It was wonderful to be given this opportunity to be here, and this is my first forum. I promise you it won't be the last. And it's been fantastic to be part of this, and I want to make full use of this time. So quickly, I'll start with what I think is the most important slide. This is from 7 o'clock this morning, and you can take multiple messages from this: host, pathogen, DBT, ICMR, whatever you want. Okay, but I think the most important thing is the last part there, which is equilibrium, and this is something that viruses also attain or hope to attain, when they infect individuals at a population level at least. So today I'm going to talk to you about an equilibrium virus, HCMV, or human cytomegalovirus, and how, I've underlined the word unifying there, how we have the virus, we have the host microbiome, that's bacteria. In this meeting we are going to talk about bacteria, yes, and obviously the immune system, of which or through which I learned my virology. So CMV is an old virus, very much present in our population and definitely at equilibrium, in the sense that in terms of the population, it doesn't cause that significant amount of pathology that severely affects the reproductive capacity of the population. At the same time, it effectively achieves almost ubiquitous transmission. And I want to talk to you specifically about its role in pregnancy, keeping in the mandate of my institute. And it is actually not very well known that almost 2% of live births worldwide and congenital infections can be attributed to HCMV. So the conundrum that I and my collaborator work on is essentially why India has a high seroprevalence of HCMV specific antibodies, especially in women who are of reproductive age. But at the same time, it is this same population that also has the highest incidence of congenital infections. So much for antibodies, right? So much for what they can provide or protection. So why does this virus elicit such a good response and yet be so successful in being maternally transmitted to children? So the specific case that I'm going to talk to you about is neonatal cholestasis, which is a pediatric infection. It is related to liver pathology. I won't spend a lot of time there, except to say that ours and other work has highlighted the importance of HCMV infection in the pathology of neonatal cholestasis. These are mainly associative studies to begin with. And in this work, what we were trying to understand is the interplay between HCMV infection and immune dysregulation that results in the severity of neonatal cholestasis. So just to be clear, this is a pediatric disease that occurs in individuals who are, in our case, we kept the age, I guess, to about 5 to 6 months post-birth. And where does the microbiota come into all of this, right? So the microbiota, I'd urge everybody to look at this paper. It's an old but highly cited paper, which actually implicated specific immune responses that were modulated by selectively deleting microbiota taxa. And this had a viral connection because it was against influenza. So taking this as an inspiration, my colleague Vikrant Bhor and I decided to probe this axis that we coined as the microbiome immune axis against viral infections and address these particular questions. Okay.
what was our study population? So it was a hospital in which we had access to infants who were suffering from neonatal cholestasis. These are our selection and exclusion criteria. This is our sample size. Of note here is that you'll see that the HCMV infected individuals were stratified into IgM negative and IgM positive individuals. Why were we interested to stratify that at that point, from that point of view? Is because it was reported that apparently the IgM positive, so the ones who had acute infection, might have had a greater pathology or a more severe pathology. So bottom line is, why is this happening in the context of acute HCMV infection, and is there any role for the microbiota or dysbiosis in the sequelae that we observe? So quickly looking at the clinical data, you can just focus on the two red boxes which highlight the two clinical parameters that were found to be significantly different or elevated in this case, suggestive of higher pathology in the IgM positive group, telling us straight away that yes, in the condition of neonatal cholestasis, which affects the liver, we are looking at two parameters that seem to be worse off when individuals have acute IgM positive infection. Jumping straight into the microbiome data, we used stool samples to generate this data, and this is just a global view of looking at what we call as alpha diversity, which is just briefly trying to figure out, or at a global level, do the microbiota or the microbiome, does it look different in our three groups? So the color coding, as you'll notice here, is, try to get this pointer going. So the healthy group that is blue, the IgM positive group that is pinkish or red, and the IgM negative group, both of which are infected, and the IgM negative group is green. So this is just a diversity analysis which is based on your 16S rRNA sequencing, and then to the right we look at another kind of diversity we call beta diversity, which is more about the qualitative differences in each of these groups, and you can see here through the PCOA plot, clearly again the groups do segregate in terms of their microbiota, suggesting that acute HCMV infection does result in a specific microbiome signature that is disparate from neonatal cholestasis background or IgM negative infection. When you try to resolve this at the level of individual taxa, we notice, so there's this ratio that's kind of important in the area of microbiome. That is the B to E ratio, that is the bifido to the entero ratio. So as you might guess, it's essentially gram-positive and gram-negative bacteria, and we're looking at the ratio of the phyla or the taxa that are gram-negative, which are the pathogenic or entero kind of bacteria, some of which include proteobacteria here, as you can see, clearly enriched in the IgM positive group, and then conversely you have more gram-positive bacteria like actinobacteria, which are reduced in the IgM positive group. If you do this even in the form of a heat map, you can clearly see a nice continuum where in the healthy group you have very enriched bifidobacteria, which is beneficial, and then you have that being completely depleted in the IgM positive group, but not so much in the IgM negative group. So moving on to how populations of bacteria would interact in the pathology of this disease. If you just start, it's a busy slide, there's no need to look at everything, but if you start from the bottom panel and go up and then to the right, which is the grouping of healthy IgM negative and the IgM positive, all the gram-negative bacteria are coded in red. All the gram-positive bacteria are coded in green. And you will see basically the red, that is the population that is enriched in the IgM positive group, is basically very underrepresented in the healthy group. And there's this almost continuum where you see more red starting from the healthy, going up to the IgM negative, and then to the IgM positive. Similarly, you'll see a drop in the green, which represent individual taxa that actually were dysbiotic in the IgM positive group. So all right, we've clearly shown that there's a dysbiotic signature. Where does the immune response come into all of this, right? So we were lucky and fortunate to sample during the course of their routine treatment blood from these individuals, and I'm quickly just going to point your attention to the CD4-CD8 ratio in the individuals who are IgM positive. So you'll notice that these guys had a clearly, so the IgM positive group is the pink, remember? So they clearly had a lower ratio expansion of CD8 T cells, depletion of CD4 T cells. And what functional relevance does this have? So if you look at within these same cells, using facts here, functional attributes of cytotoxic potential. So granzyme B positivity within these same cells, you'll notice pretty much ubiquitously, whether it's T cells here or whether it's NK cells at the bottom here, the group that is IgM positive had increased cytotoxic potential in terms of intracellular granzyme B. Now the color coding is such that the individuals who are not black are the ones where matching 16S RNA data did exist. So we could do a direct correlation between the immune parameters that we observed and the dysbiosis that we observed in these same individuals. So just moving quickly to that, you'll see here, this is basically just a Spearman correlation, a non-parametric. Everything is significant here except for the gradation that I want to highlight. So if you start with the pale yellow, these are negative correlations, and then moving up to the red, which are positive correlations. So Firmicutes, which is a beneficial bacteria, has a nice strong correlation with CD4-CD8 ratio, as you would expect, and then a negative correlation with cytotoxic potential in the NK cell. Now you notice that the NKT granzyme B was elevated in the IgM positive population. Similarly, if you look at gamma protobacteria, gram-negative, LPS producing bacteria, you have a nice strong positive correlation with increasing gamma proteobacteria observed in the IgM positive group and increasing NK granzyme B. Similarly, as you go down, Fingoldia, which is again a beneficial bacteria, nice strong correlation with CD4-CD8, and then really low inverse correlations with cytotoxic T cells and NK cells. So this suggests that we were able to finally correlate here an immune phenotype with dysbiosis in a particular condition. When we try to understand how much we could mechanistically link this, we were successful in looking at various mediators, including inflammatory mediators, including LPS, bacterial products. And again, when we put all this together in a PCOA, we were able to segregate these groups, suggesting there is an integrated gut microbiome immune axis. I'm going to end just by showing you what we believe is going on. This is what's happening in healthy infants. You have homeostatic production of good bacteria, less pathogenic bacteria. Because of that, less LPS, less breaking of gut integrity, and because of that, you don't have systemic inflammation. But when you go to HCMV infected infants, especially those who are IgM positive, you have a pathogenic consortium that overgrows the beneficial microbiome. You obviously then have reduction in the short chain fatty acids of butyrate and acetate, increased LPS, more barrier, or I should say microbial translocation that occurs, which drives systemic immune inflammation in term possible pathology. So with that, I'd like to end. We don't spend all of our time in the lab, as you can see here. And of course, I'd like to thank Vikrant Bhor, my buddy and collaborator in this project, and of course the core study team. Thank you very much for your attention.
Thank you, Dr. Bhasin. We have time for one or perhaps two questions. Not then, I'll ask one quick question. It's all to natural birth or to C-section.
Yeah. So our group, our study group essentially had an equal distribution of both. We matched it because we had, access to when these kids were undergoing surgery. So at that time, we matched the individuals in our group for two things. Both was the modality of birth and also whether they were breastfed or not. So we included that in our stratification.
Yeah, not from the field, but there is general consensus that natural birth provides the optimal microbiota, which is helpful in preventing early infections.
So the very first microbiota, if you have vaginal birth, you're gonna acquire is your mom's vaginal microbiota. The thing is, why does what is CMV doing to in these kids perturb that balance? And there's other stuff we can talk about later, but clearly there is something that is pushing dysbiosis.
All right, thanks very much.
All right, thank you.
Join our hands to.
Thank you.
Next speaker is Professor Abid Saha. He comes from Presidency University now, but he's been trained for his PhD in Bose Institute, then for postdoc in University of Pennsylvania, working on DNA viruses. He's been around in fellow and a secondary fellow as well. And hearing about some DNA virus from him.
Thank you, Shashank, for the nice introduction. Thank you, Soumitra, for squeeze me in. So the title of the talk, as you can see, the differential carbonic anhydrase activities control EBV-induced B cell transformation, as well as the lytic cycle reactivation. So EBV, as we all know, that the first cancer virus discovered is associated with approximately 1% of the total cancers worldwide, mostly the B cell lymphoma, like Burkitt's lymphoma, Hodgkin's, DLBCL, and immunoblastic lymphoma. Like all other herpes viruses, EBV also has biphasic life cycle, like latent phase and the lytic phase. So after initial infection on epithelial cells, oropharyngeal epithelial cells, EBV primarily infects the naive B cells, and the naive B cells are growth transformed. So EBV actually resides for the in the memory B cells for the rest of the life of the host, but intermittently it actually comes back to the lytic replications, when the B cells transition into the plasma cell differentiation. And this lytic cycle replications requires for the maintenance of the viral progeny, where the latent infections are actually associated with the cancers listed here. So to understand the EBV biology or pathogenesis, particularly in the B cells, we EBV biologists have a very nice in vitro system where the PBMCs are infected with the EBV in vitro, and within a time span of 1 month, these B cells are growth transformed into a continuously proliferating lymphoblastoid cell lines, known as also LCLs. And for the lytic cycle replications, there are a lot of chemical reagents, like a combination of formol or sodium butyrate, as well as proteasome inhibitor or antibody cross-linking that activates EBV lytic cycle replication. So we focus in these two windows. One is the lytic replication, another is this hyperproliferation, which is actually present in the early latent infection stage, 2 to 4 days. As the study suggests, during this phase, EBV profoundly modulates the B cell architecture, which requires for this the followed B cell proliferation and then B cell lymphoma development. So to understand what are the B cell factors required for this EBV pathogenesis or the EBV induced B cell transformation, so we did a simple global transcriptomic analysis for these two windows, and we found a very interesting data. We found a carbonic anhydrase 9 CA9, which is transcriptionally activated during the latent infection, while during the lytic replication mode, it actually transcriptionally suppressed. So it is differentially expressed. So what does this carbonic anhydrase do? So far, there are 12 catalytically human carbonic anhydrase isoforms have been identified, and all these carbonic anhydrase. So some of are localized, is distributed in the cell, in the membrane, in the cytoplasm, as well as in the mitochondria. So all of these are required for the maintenance of the pH homeostasis, both the intercellular pH as well as the extracellular pH. Among these all isoforms, CA9 is actually been associated with the poor prognosis of many solid cancers. However, the no information or the sparse information are available for in case of lymphoma and leukemia. So for that we started our experiment, and first we tried to validate our RNA-seq data by qPCR, which is not shown here, and Western blot analysis, as well as by immunofluorescence study. And we also extended our data in lymphoblast cell lines. So we used multiple donors in case of both EBV transformed as well as primary infection model. All that that showed there is a upregulation of or transcriptional activation of this particular protein. So it's not only in the primary infection model, but as well as in the transformed cells. So we showed like, this, the only the membrane associated carbonic anhydrase 9, CA9 along with the CA12 are only expressed. So no other carbonic anhydrase, we did not observe like, any other carbonic anhydrase expression. So GTEx analysis showed that not the CA12 but the CA9 is associated, it's highly elevated like, in EBV transformed cell. So whether that is true or not, so we did a carbonic anhydrase assay and like, using subcellular fractionation in both cell lines, like, transformed cell lines, and we actually like, found the carbonic anhydrase activity is associated with the membrane fraction and not in other fractions. So this elevated carbonic anhydrase expression as well as carbonic anhydrase activity, whether it's required for this B cell transformation or like, LCL's outgrowth. So we use like, utilize two different strategy. One, like, we use like, there are several carbonic anhydrase inhibitors, both in the clinical trial or like, FDA approved, and like, definitely like, shRNA mediated knockdown of CA9. So this is the picture that shows that how EBV, that is a GFP tag, that it transforms the B cells within a period of 28 days. However, in the presence of like, the carbonic anhydrase inhibitor, there is like EBV failed to transform B lymphocytes. So when we checked the intracellular pH as well as like the carbonic anhydrase activity, and it shows there is a peak like, at like, the 2 to 4 days time point, that hyperproliferation stage, but whereas like, this using the carbonic anhydrase activity inhibitor, that it actually blocks both the activity as well as the intercellular pH or accumulates of the protons in the intercellular milieu. So in case of the transformed cells, so the decrease of carbonic anhydrase activity by using like, this is the data for only one inhibitor. So we have, as I said, we use like, three inhibitors together, and so decrease in the carbonic anhydrase activity, which is associated with the decrease in the intercellular pH, that means more acidic pH, associated with the decrease in the cell viability as well as the less colony formation ability of the transformed cell. So the carbonic anhydrase inhibitors that causes like apoptotic cell death, and by using Western blot as well as real-time PCR, we show that the apoptosis is p53 mediated, but we actually like, found a very interesting data that is the inhibitor is not actually like, blocking the enzymatic activity, it's actually blocking the expression of the protein. And it's basically at the transcriptional level, which we confirmed by the qPCR, the mail cup analysis as well as the semi-quantitative PCR. As I already mentioned, I'm not showing you the data, that using shRNA mediated knockdown of the CA9, that we showed that CA9 expression is actually required for the EBV transformed cell. I will go like, quickly. So for the finding the mechanism, so we checked like, the ChIP-seq analysis, and we found that only EBNA2, that's the fast viral latent protein like, expressed, along with many B cell specific transcription factor that co-localize on the CA9 promoter or the enhancer region. We confirmed by the qPCR analysis that RBP J-kappa2 is actually the main protein, that is an adapter protein for the EBNA2 across the human genome. So we use like an EBNA2 deleted virus, and that confirms that yes, EBNA2 is the culprit like, for the CA9 transcriptional activation, but we don't know what is happening in the lytic replication. For that, like, we need to know that BGLF1 is the first protein to be expressed during the lytic replications, which actually responsible for the activation of more than 40 lytic proteins responsible for the complete lytic replication of the viruses. And we confirmed like, our RNA-seq data again by the Western blot, qPCR and CA activity, and whether BGLF1 is responsible for this downregulation of the CA9. We used our exogenous system, and we showed like, in a dose dependent manner that BGLF1 is transcriptionally downregulated the CA9 expression. So whether BCLF1 belongs to an AP1 transcription factor family, so using cheap qPCR we found BCLF1 binds to the proximal region of the CA9 promoter region is like very different than the EBNA2 where EBNA2 binds. And using cheap qPCR analysis we found like the BCLF1 binds to during the lytic replication onto the CA9 promoter region. And by using the more truncations and point mutation, we confirmed yes, BCLF1 using the TAG domain which is required for the chromatin accessibility that requires for the transcriptional repression of the CA9. So this is the final model where we showed the EBNA2 which requires for the CA9 upregulation and the pH homeostasis which is required for the B cell transformation as well as L cell outgrowth. Whereas during the lytic reactivation, BCLF1 express which downregulates the CA9, but we do not know whether this accumulation of the protons which requires for it acts as a prerequisite for the viral lytic reactivation. So that's where I will end. So I will acknowledge the contributor of the work, my collaborator, and the work is done by Somoresh mainly. And this is my lab. This is the two campus where I belong. So during the lockdown period I moved my lab from the old campus, the 200-year-old campus from the new campus at Newtown and the funders as well. Thank you.
Thank you, Ravi. In case of time, we have one or two questions.
I have a very quick question.
Yeah.
Have you ruled out the involvement of enhanced RNA in this loop?
So it's possible, but I did not rule out that thing. Yeah, because EVV, as you know, there are a lot of enhancer RNA there. There is a high possibility because EBNA2 is also involved there.
Questions from the audience? Now let's thank again Dr. Saab.
Thank you.
We'll invite our next speaker, Dr. Ghulam Sayed, from IRS Bhubaneswar. He trained for his PhD in University of Hyderabad, then went to University of San Diego for his postdoctoral training, came back to India with the Ramalinga Fellowship, and also welcome to Single Alliance Intermediate Fellowship. This is into Dr. Ghulam Sayed.
Just check if the pointer is working. The slides are moving.
Yeah. Good afternoon. Thanks for the organizers to squeeze me in and give me the opportunity to attend this wonderful meeting. It's good to be with the colleagues and spend some valuable time with them. So today I'm going to talk about hepatitis C and the endomembrane remodeling. Basically, my lab works on the virus host interactions of flaviviruses and hepaciviruses. And in addition to this, also look into the molecular processes which aid in the viral dissemination to identify host factors that act as in proviral manner. So in this context, I'm today going to show my work on hepatitis C virus secretion. Hepatitis C virus, as you all know, is a chronic hepatitis, causes chronic hepatitis and leads to end-stage liver diseases like liver fibrosis, cirrhosis, and hepatocellular carcinoma. So just to keep in time, let's skip this introduction. And it's a +N single-stranded RNA virus, and it encodes a single-stranded polyprotein that is processed into 3 structural and 7 non-structural proteins. And then the 3 structural proteins become a part of the virus, whereas the non-structural proteins take care of the other aspects of the viral life cycle. So there's a lot known about hepatitis C virus. It's, I think, second most studied virus apart from HIV. There's good knowledge about entry, translation, replication, as well as the assembly of the virus. But how does the virus secrete? And what is the mechanism or route that it traverses to egress the virion particle is not very well addressed. So we've been working on this aspect for quite some time, and we have shown that HCV secretes through the COP2 vesicles through EM imaging, immunogold, immunolabeling. We have clearly shown that the virus particle is engulfed in COP2 vesicles and then secretes out through the from the ER in COP2 vesicles, which then suggests that we, in this context, we ask the question like, how does the virus affect the global protein secretion? How does it exploit the ER exit sites and the ER Golgi intermediate compartments? And are there any COP2 coat proteins that specifically required for the release of the hepatitis C virus? Because the COP2 inner coat proteins are involved in cargo selection and recruitment to the site of COP2 biogenesis. So we used hepatitis C infection in the hepatoma cell line HUH7, and we came up with an MOIF3 that gives about 80 to 90% infected cells at 72 hours post-infection, and this is what we use in the rest of the study. First thing we asked is how HCV affects the global protein secretion, and we used SSHRP reporter, which actually is a secretable form of horse radix peroxidase. And if you look into the horse radix peroxidase activity in the culture supernatant, you will be able to figure out the ER Golgi conventional secretory pathway. And using this reporter, we've seen that HCV significantly inhibits the secretion, global protein secretion, and there's an accumulation of the SSHRP flag in the lysates, as well as we've seen shown that there's also a tremendous accumulation of the protein in the ER exit sites. So then we went ahead and looked into the liver-specific proteins that are secreted through HCV, like transferrin, albumin, and ApoE. We also saw that these are also inhibited, the secretion of these proteins is inhibited upon HCV infection. And we also found that the intracellular level of these proteins are also downregulated in HCV infection. And further investigation, we found that actually these proteins are taken up or eaten through the autophagic process, and inhibiting in the autophagic process led to their accumulation, suggesting that HCV not only reduces the secretion but also utilizes them or through the autophagic process. So we then asked the question how HCV alters the organization or the function of the early secretory components, the ERES and ERGIC. So we use certain markers like Sec16, ERGIC, and 53 and TFG. Sec16 is actually a protein that it enriches in the ER exit sites. These are basically extensions of the ER where the COP2 biogenesis happens and cargo recruitment happens. And then TFG is a protein that actually helps in the formation of a higher level ER, transitional ER, that promotes the secretion of large proteins like collagen. Whereas ERGIC 53 is a part of the ER Golgi intermediate compartment and involved in the sorting of the glycoproteins. So we use these markers, and we also tried to figure out how HCV utilizes these proteins in its life cycle. So looking at the morphology of these components, in the mock cells you can see that the ERGIC or the compartment as well as the ER exit site compartment is nicely distributed in the perinuclear region across the cell, whereas in case of the HCV infected cells, we found that they are more compact and more less in distribution in the total cell. And when we analyze these images for the overall expression, we seen that there is a reduction, as well as when we analyze their ratio with respect to the total cellular area, we see that the ratio increases in the infected cells, suggesting that during the infection, the endomembrane compartments occupy lesser cellular space. So we also looked into the other protein, the TRK fuse genes, because this was important for us because it controls the export of large cargo. And HCV is actually a unique cargo because it's unlike other viruses. It's about 100 nanometers in size, and that's why it requires a lot of COP2 remodeling to ensure its secretion. So when we looked into TFG localization in HCV infected cells, this is how it looks in a normal mock cell. And this is what happens in case of HCV infected cells. We see its enrichment in the ER exit site and a lot of enhanced colocalization with the ER exit site marker as well as the ERGIC compartment. So we then went ahead and tried to see what is their role in the life cycle of the hepatitis C virus. But before I touch upon that, I also want to show you that HCV also induces Golgi fragmentation, and so with the increase in time post-infection, you see an increase in the number of cells that display fragmented Golgi. So here we are seeing infection that promotes compactness of the ER Golgi intermediate compartment, ER exit site, as well as the fragmentation of the Golgi apparatus, which infers that it definitely has a negative effect on the global protein secretion. We use different kinds of systems, pseudoviruses, subgenomic replicons, and also a complete viral life cycle representing cell culture model to identify the role of these proteins in the life cycle. Initially, we started off with depletion of ERGIC53 to siRNA-mediated silencing, and what we realized that ERGIC53 knockdown inhibited the viral entry, and the knockdown also was responsible for reduction in the putative viral receptors like claudin-1 and occludin, the tight junction proteins that are known to promote the viral entry. However, in case of Sec16 knockdown, we have seen that it doesn't affect the entry, but it does affect the replication, and this we have done with the subgenomic replicon, whereas ERGIC53 knockdown or the TRK fused gene knockdown does not affect replication. Then we found that overexpression or rescuing the Sec16 knockdown is able to rescue HCV replication in the replicon cells, and we later on tried to figure out why would Sec16 knockdown specifically affect replication, whereas in case of TFG and ERGIC, it's more about entry and release. So we later on tried to see, because ERGIC and the ER exit sites contribute to the biogenesis of the autophagy vesicles. So we found out that the knockdown of Sec16 particularly affects the biogenesis or initiation of autophagy. Then, so let's keep that. We use this life cycle, the cell culture model representing the complete life cycle of the virus, and using knockdown of the ERES and ERGIC components prior to infection and post infection, we came to figure out that in case of Sec16 and ERGIC knockdown, there is no significant effect on the intracellular viral RNA, but TFG knockdown leads to an increase in the intracellular viral RNA as well as the intracellular viral titers, which suggests that there is a reduction in the secretion of the virus, and TFG leads to, promotes the release of hepatitis C virus. That's what we have seen, that the extracellular virus was exceptionally low in TFG knockout, as well as the extracellular viral RNA. ERGIC is also low, but this is because of the reduction in the NT, not because of the effect on the viral secretion. So expression of the Sec16 rescued HCV replication in the cell culture model, as well as the expression of TFG in the knockout cells or knockdown cells rescued HCV release in case of the HCV infected cells silence for TFG. So this suggests that Sec16 plays a role in replication of the virus, whereas TFG is specifically required for the hepatitis C virus secretion. So we then used a compound that inhibits the intra-Golgi transport, FLI06, and using this compound we have significantly seen its effect on the release of the virus, further suggesting that the ER Golgi pathway plays an important role in hepatitis C virus release. So the overall summary is that in case of normal cell, you have a nicely distributed ER exit sites and the ER Golgi intermediate compartments, and the process of autophagy seemingly goes on without any issue. Whereas in case of the HCV infected cells, you have more compact ER exit sites and ER Golgi intermediate compartments, and you have more number of autophagosomes, because HCV is known to inhibit the fusion of the autophagic vesicles with the lysosomes. And this is how HCV exploits this endomembrane system for its own secretion. We are clear about this aspect, but we are still yet to work out the crosstalk between the ER exit sites and the autophagy and its role in HCV secretion. Because this is the phenotype that you also see in case of starved cell, nutrient starved cells. You have compact ERES and compact ER-Golgi intermediate compartments. So, thanks for the. Okay. So this is the conclusions, like knockdown of Sec16A inhibits the virus replication in assembly, whereas knockdown of ERGIC53 significantly inhibits the viral entry. And interestingly, knockdown of TFG, which is also an ER exit site protein, selectively inhibits only hepatitis C virus secretion. So finally, I would like to acknowledge my group, particularly Kiran, who has done this work, and Bharati, and then the funding from Indian Alliance and SERB and PBT. Thank you.
Yeah.
Yes, sir.
Wonderful work. See, just tell me, this is a technical question. The TFG level in HbH7 and HG2, are they less compared to 7.5?
No, sir.
But they are deficient in virus release.
Yeah, if you're knocking them down, they're deficient in virus.
No, I'm saying some of the cell types.
Yes.
Virus release is affected. I was just wondering if you can check the level of TFG there.
7.5 cells are defective in the RLR pathway.
Right.
That is why they have more susceptibility to the infection, not related to, and they are defective in lipid metabolism also. So it's not a good idea to use them for secretion aspect. The 7.5 and 5.1 cells. Yeah.
Any questions from the audience? Now, a quick question.
Yeah.
This is a phenotype of actively infected cells. What happens in a chronic infected liver? The secretion function is.
Yeah, in case of human samples, what you see is steatosis. There's enhanced lipogenesis, but less release of the lipoproteins. That leads to steatosis or NASH kind of clinical manifestation in the infected cells. So I believe that the overall effect on the global secretion may be leading to this kind of clinical manifestations.
Thank you, Shubha.
Thank you.
Next student speaker is Amrita Ray, coming from the same institute. I just Bhubaneswar, working with Dr. Soma Chattopadhyay. She's going to talk about combinatorial therapy for chikungunya.
Yeah. Just try moving it. Just try moving the slides.
Good afternoon, everybody. I'm Amrita Rai, currently working under Dr. Soma Chattopadhyay from the Institute of Life Sciences. So, thank you very much for allowing me for the short talk on the topic DDA-BT1, a combinatorial therapeutic against chikungunya infection and inflammation. As is known to everybody here, that chikungunya virus is an arthropod-borne alpha virus belonging to the Togaviridae family, and of all the symptoms, debilitating arthritis is the major one. Its genome structure has two open reading frames which encode for the non-structural and structural proteins respectively. The virus has been now currently identified in around 40 countries and is referred to as a category C pathogen. So a lot of study is going on different stages of the viral life cycle, but still there are no licensed antiviral drugs available, only symptomatic treatment is present. The rationale of our study is there is no licensed antiviral available. Inflammation is one of the major symptoms, so they are treated by NSAIDs. Activation of angiotensin II receptor type 1 plays a key role in the progression of inflammation. Telmisartan is a known selective AT1 blocker, and in our lab it has already been shown that telmisartan inhibited chikungunya infection in vitro as well as in vivo. There is another compound, salicylic acid, which also inhibits cyclooxygenase to mediate anti-inflammatory action. So it was thought worthwhile to design a molecule to achieve a higher antiviral as well as anti-inflammatory efficacy. So DDA-BT1 was synthesized as a conjugate of salicylic acid and telmisartan. Its molecular weight Its molecular weight is around 634 Dalton and purity of 96.5%. This work was done in SOA University by Dr. Subudhi. So first we had to see whether DDABT1 was more effective than the parent compounds or not. For this we performed the MTT assay and observed that the CC50 was around 700 micromolar, and as you can see the plaque assay very efficiently shows that DDABT1 inhibited viral infection compared to the parent compounds. So then we had to check the IC50 of the drug, and we saw that at 0.1 MOI the IC50 was 21.07, which is much better than that of telmisartan, which is 41.2 micromolar at 0.1 MOI. So now we had to see at which phase was DDABT1 acting. For this, we added drug at 3 different stages, that is 1 hour prior to infection, during infection and post-infection. And as you can see, the CPE images clearly indicate that the drug worked better when added post-infection, which was corroborated by our plaque assay data, which showed 75% reduction in plaque assay when added post-infection. We also checked the viral proteins. Non-structural protein, NSP2 and E2, and we saw that there was considerable fall in the protein level when the drug was given in the post condition, which was again corroborated by our immunofluorescence data. Then we had to see exactly post entry at which stage the drug was acting. So for this, we performed a time kinetics and we observed that DDABT1 inhibited chickpea infection throughout, more prominently in the early stages of infection. So now we had to see whether DDABT1 mimicked the known mechanism of telmisartan, the parent compound telmisartan, that is AT1 inhibition, which has already been seen in our lab, that telmisartan inhibited chickpea infection by inhibiting AT1. So for that we treated the mock cells with the drug and we observed that there was non-significant changes in the AT1 expression. Then they were infected and treated with the drug, and it was seen that just like telmisartan, the AT1 expression infection went down, and AT1 so was the AT1 expression. Similarly, in case of DDABT1, the infection went down and also the AT1 expression. Then we added a compound called AG, which augments infection as an agonist of AT1. Even after addition of AG, it was seen that DDABT1 inhibited infection considerably. So DDABT1 retains the ability of telmisartan to inhibit AT1. So as it is, we are still undergoing a lot of studies in this mechanism aspect, and we have to also do the antiviral and antiviral study in vivo, but we have already proceeded with the anti-inflammatory to check the anti-inflammatory property of the compound in vivo. So it was first seen in Wistar albino rats. And an acute toxicity, oral toxicity study was done, and was seen that it was non-toxic up to 2000 mg per kg. The paw edema was first the rats were treated with the drug, and 1 hour after that 1% carrageenan was induced to induce the paw edema, and at 1, 2, 3 and 4 hours the paw edema was measured, and it was seen that DDABT1 inhibited paw edema considerably compared to the parent compounds. A cotton induced granuloma was done, and it was seen the wet weight and the dry weight of the cotton pellet was taken, and the drugs were added for 7 days continuously, and it was seen that the exudate and granuloma weight went down considerably when the rats were treated with DDABT1. So DDABT1 had a better anti-inflammatory property and also reduced acute inflammation than the parent compounds. Now we had to check whether DDABT1 reduced chronic inflammation as well. So for this again, a CFA-induced arthritis model was taken, and the drugs were added on 7th, 14th, 21st and the 28th day, and the change in paw volume was measured, and was seen that DDABT1 decreased the paw volume considerably when the rats were treated with DDABT1. The X-ray images also clearly show that the inflammation was very much reduced when the rats were treated with DDABT1. The other inflammatory factors, that is the erythrocyte sedimentation rate and the rheumatoid factor, were also checked, and it was also seen that it was reduced considerably in case of DDABT1. So DDABT1 not only retained the anti-inflammatory property but also is better than the parent compounds. So I would like to conclude by saying that DDABT1 inhibits chickpea infection better than telmisartan salicylic acid, their combination in vitro, and it had a CC50 concentration of around 700 micromolar. It had IC50 better than that of telmisartan. It inhibited chickpea infection and reduction in chickpea protein levels when it was added post-infection, and it inhibits predominantly the early phases of the chickpea life cycle. It also retains the ability of the telmisartan to inhibit AT1 and has better anti-inflammatory properties than the parent compound. Our future work would be to study the mechanism of action and also to study its anti-chickpea efficacy in vivo. So I would like to thank our director ILS, my guide Dr. Soma Chattopadhyay, Dr. B.B. Subudhi, our collaborator who has actually synthesized and characterized the drug, my seniors Dr. Prabhudatta and Dr. Saikat, UGC for my fellowship and RCB for registration, my lab mates and friends. Thank you.
Thanks, Amrita, for keeping it on time. Any questions from the audience?
Did you by any chance try the combination of DDABT1 along with telmisartan? Like both TM plus DDABT1?
Oh, separately, you want to say that? I will.
TM has shown anti-chickpea activity and DDABT1 also has anti-chickpea activity.
Actually, DDABT1 is a conjugate of telmisartan and salicylic acid. It is synthesized as a single compound. So it already has telmisartan in it.
Alright, so is the virus difficult or antivirals are not that great? Because the inhibition is very poor. It's just few fold reduction in PFUs. With conventional antivirals, that's nowhere. Not easy.
Sir, I would say that maybe we are not gone to the other level of expertise of I have not gone to the other level of expertise of commenting on that. But I would say that although lot of antiviral studies are going on, you can see that certain drugs are working better in vitro condition, but when we are taking it to in vivo model, they are not working properly. So we still have to do with the in vivo studies, and we want to see if it is actually working better, then we would definitely want to take it forward. And further, it has to be taken up by the pharmacological companies also to proceed with the study. Maybe that is where we are lacking.
So there is still room for SAR studies and further improvement of the need. That's what I mean.
Yes, sir.
Right, thank you.
Thank you, sir.
Thanks, Amrita. Next speaker is Ms. Deepa Singh from Krishna Inampuri Lab at AIIMS, New Delhi. She is going to talk about structural aspects of SARS-CoV-2, RDRP.
So, good evening to everyone. I am Deepa Singh, a PhD scholar from AIIMS New Delhi. I am pursuing my PhD under Dr. Krishna Kishore Inampudi. And first of all, I would like to thank to the organizers to give me this opportunity to present my work today here. And this is the title on which I am going to talk. Interaction of SARS-CoV-2 NSP7 or NSP8 along with NSP12 cause constriction in the RNA entry channel. We all are aware from the SARS-CoV-2 virus and which was major responsible for the COVID-19 pandemic. If you see the If you see the structure of the virus, it's contain the positive sense single stranded RNA and its open reading frame code the structural and the non-structural protein. The non-structural protein in which NSP12 is majorly responsible for RDRP activity. If you see the structure of the NSP12 in the N-terminal domain, it contains the neuron domain which interact with the C-terminal by the help of interference, and RDRP activity of NSP12 is majorly present on the C-terminal domain, which contain 3 domain: finger domain, palm domain and thumb domain. This NSP12 require 2 other accessories protein for their RDRP activity, that is NSP7 and NSP8, and it make the RDRP holoenzyme complex. So how these 2 protein interact with NSP12? The 1 unit of NSP7 and NSP8B interact with the thumb domain of NSP12, and the 1 unit NSP8A interact with the finger domain. The template and primer RNA duplex complex bind with the palm domain, which have the main catalytic sequence of the RDRP activity. We have lots of abundant information about the RDRP, but so let's shed some light on the background of RDRP work. Till now it has reported that this RDRP activity of NSP12 is present only in the presence of 7 and 8 protein, but there was one contradictory data which has shown that NSP12 alone can also bind with RNA. If we compare the SARS-CoV-2 polymerase activity with SARS-CoV-1, so in 2014 it was reported that this SARS-CoV-1 NSP12 activity is also present in the presence of this 8 and 7, but there was also one contradictory data which shown that NSP12 alone can also active. So that time my question was, if this NSP12 of SARS-CoV-1 and SARS-CoV-2 had 96% genetic sequence similarity, so why this was showing that much functional difference? So there is lots of contradicting data which need to be understood. So for that I characterize the RDRP complex with using the different RNA templates, and I also investigate the individual contribution of NSP12 and NSP7 and NSP8 protein, the role of RDRP holoenzyme complex formation. For that I purified all these 3 protein in the bacterial expression system and confirmed by the SDS. This purified protein I used in vitro and reconstitute and make the RDRP holoenzyme. After that I need to check this RDRP holoenzyme at the what concentration it was binding to the RNA. For that I perform the protein RNA interaction by the IMSA, electrophoretic mobility shift assay. For that one I use the following RNA templates which I synthesized and the purified, and the following fluorescent primer I use it and prepare the duplex. This duplex at the constant concentration I incubated with RDRP holoenzyme at different concentration and check the binding, and I quantify all the bands which was bound with the RNA. So from that I concluded that at the 10 micromolar concentration this was able to bind with the RNA, and this concentration I used for the further study. After that I need to check the activity of this RDRP complex. For that I perform the fluorescent extension assay, and this template duplex I use it and incubated with different combination of NSP protein. In NSP protein combination I took RDRP holoenzyme, NSP12, NSP12 plus 8 complex and NSP12 plus 7 complex. What I found in the results, the RDRP holoenzyme was able to extend the RNA which already reported. Surprisingly, we also got the extension when the NSP12 alone was present, and these 2 complex was not working. So that time I was surprised, like previously it has reported that this NSP12 alone is not active. So for that one I again design and synthesize and purify all these following templates and check the extension activity further, and we did not get any significant changes in the polymerase activity. So I compared my data from the previous reported study, and I found that in the previous study people have used the RNA loop template. So I designed that and ordered and performed the fluorescent primer extension assay, and I found that this RDRP holoenzyme was able to extend this RNA loop template, but when NSP12 alone was present it was not showing that much extension activity, and the activity was very less, it was less than 20%. So maybe that RNA loop template was not a good model for this study. We also checked the DNA dependent DNA polymerase activity of RDRP, and following templates we used for that one, and we checked the similar kind of condition of experiment we have taken, and we found that this RDRP was holoenzyme was also able to extend the DNA template as well. So the presence of this cofactor NSP7 and NSP8 facilitated the necessary conformation flexibility for the DNA polymerization by NSP12. So this is the overall experiment result of primer extension assay. So you can see when we use the linear template loop and the DNA template, the RNA holoenzyme was able to extend the RNA, but NSP12 condition only linear template was showing the extension activity, and these two complex was not able to extend the RNA. So I had doubt why this NSP12 alone can synthesize RNA, and it is, if it's working alone, so why these two complex NSP12 plus 7 and NSP12 plus 8 is not working. And our next question was, does the RNA duplex bind to the NSP12 in the pairwise combination? So to check that one, what we did, we have taken this NSP12 plus 8 and NSP12 plus 7 complexes, and we further did the protein interaction study. For that, we checked that one, we use only RNA template as a control, and we check the binding only in the NSP12 also, which we have used as a positive control, and we got the binding. When we use these three NSP's protein together, and we found the binding, which is RDRP holoenzyme. But when this complex was present, NSP12 plus 8 and NSP12 plus 7, we did not get any binding. So does it mean that this NSP7 or 8 was not allowing the RNA to bind with NSP12? So for that one, we further did interaction study by using this RNA loop and linear templates to check that this activity was primer template dependent or not. In this experiment, we have taken the two conditions. The first, we took the NSP12 and we added NSP7 and 8 and making the complex, and later on we added the RNA, and we saw there was no binding. The another condition was we prepare the NSP12 and RNA duplex complex, and after that we added the NSP8 and 7, and we saw that the pre-bound RNA was dissociating. So from this study, we concluded that maybe NSP7 or NSP8 in the complex with NSP12 either preventing RNA binding or it was releasing the pre-bound RNA from the NSP12. So for that, we did the kinetics study of association and dissociation. This study was time dependent, and what we did, we have taken the NSP12 and incubated for 30 minutes, and later we added NSP7, NSP8, and this complex we also used together, NSP7 plus 8 complex as well. So what we found in this result, as the time period was increasing, the primer and the RNA duplex complex was dissociating from the NSP12, and the same results we found NSP12 plus 8 combination. But when we use these two proteins, 7 plus 8 together, so it was making the RDRP complex, and we got the shifting. And another result we got it from this, the NSP12 plus 7, 8 rate of dissociation was higher as compared to the NSP12 plus 7. And we did the protein interaction study, which was supporting this data. After that, we want to know what is the mechanism of NSP7 or 8 and how it interacting with NSP12 for stopping its function. And we plan to do the computational analysis study. For that, we need to know what is the complex ratio of these two proteins. For that one, we performed the cross-linking assay, and we found that RDRP holoenzyme was actually coming at the exact molecular weight. It was showing the 1:1 ratio. And NSP12 plus 8 was showing the 146 kDa molecular weight, which was showing that the ratio of these two protein was 1:2, and the ratio of NSP12 was 7 was 1:1. This same ratio we use for the computational analysis study. So in the computational analysis study, we designed the 4 atomistic model, RDRP complex and NSP12 we use as a control, and we check the conformational changes in this complex, and we found that you can see in the initial, it is the initial conformation, how the NSP8B, NSP8 was binding to the NSP12, and the final conformation after MDA simulation, we checked that there was conformational changes was happening in the structure, which was causing the construction in the RNA entry channel. The same thing we observed in the NSP12+7 complex as well. Then we measure the average cavity volume of each complexes, and we found that RDRP holoenzyme optimal cavity volume was that much, and in the NSP12 alone it was that much, and it was dispersed the cavity because 7-8 was not there. But in the NSP12+7 and NSP12-8, it was causing the constriction in the RNA entry channel. So maybe that's the reason it was this complex was not allowing the RNA binding to the NSP12. So these are the conclusions from my study, that NSP12 alone can exhibit the polymerase activity, but this activity was very less in case of RNA loop template. And this RDRP complex was also showing the DNA dependent DNA polymerase activity, which we are reporting first time this thing. And in silico study, we observed that the constriction was happening in these complexes. These are the references, and I acknowledge to all the co-authors of this work, and my KKI lab is thankful for the DST, DBT and CSR for the funding, and thank you for your attention.
Thank you. Questions from the audience? Yes, sir.
Yes, sir.
You didn't do a DNO initiation assay, this was all primary extension.
Yes, sir, that is that one.
Yeah, but didn't you do a de novo initiation, like whether the RNA begins from de novo?
Yes, sir. I started that this sequence. I have taken the 5' of UTR of the coronavirus, and
that is fine. So you have used a primer extension, right? The enzyme can also do de novo initiation.
yes, sir, it can do.
Because in the virus, there is no primer. Who gives the primer for the virus? So it has to synthesize de novo, right?
Yes, sir. Actually, in the SARS-CoV-1 also, it has reported that 7+8 complex also work as a primase. So maybe it is assumed that in case of SARS-CoV-2 as well.
Maybe that is the reason you are seeing the discrepancy. If you do a de novo initiation, then NSP12 holoenzyme is not sufficient. Anyway,
Okay.
that's a comment.
Okay.
Thank you. For your time. Let's do a round of applause for Deepak. Last speaker of this session and this meeting also, our own Ashish Shaneja from the Samudra Das's lab, who is going to talk about the role of microRNAs in DNA pathogenesis.
So, yeah. Hello everyone, my name is Ashish Shaneja. I am a PhD student from Professor Das lab, and today I'll be talking about my work, which is an interplay between host and viral miRNAs, that is a novel mechanism for dengue virus pathogenesis. So just a brief introduction of dengue virus life cycle. We all are aware about the virus and the growing cause of concern. So dengue virus has 4 different serotypes, but the thing which I want to highlight over here is the role of 5' and 3' UTR. So these are the untranslated regions which flank the genome of the virus, and they are known to bind to a lot of factors which can cause pathogenesis. So just a brief introduction of the virus life cycle. The virus enters inside the host, replicates, translates, and then egresses out. But a very prominent part takes place inside the viral genome replication. That is the spot where the virus interferes with a lot of RNA binding proteins. So essentially these RBPs interact with the UTRs of the virus. Now one of the most studied RBP, or a so-called RNA degrading enzyme, is host hexoribonuclease XRN1. So it has been shown then that this host hexoribonuclease moves from 5' to 3' side, it encounters these resistant structures called stem loops. Now recent reports show that these stem loops, when acted by the Drosha and Dicer enzyme of the host, can give rise to small miRNA-like species called viral miRNAs. Now, in case of dengue, there are potential 6 candidates from these different resistant structures which can be encoded as microRNAs. So, also we would like to highlight the specific role of 3' UTR. It is a very structured element, and in case of dengue, the very special part of this 3' UTR is the presence of two pseudoknot structures called XrRNA1 and XrRNA2. Now these pseudoknot structures are very important and critical because they have been shown to play a host specific adaptation. And what I mean by that is that when the virus propagates in a mosquito host, essentially there are mutants which does not allow the pseudoknot to form. And when the virus shuffles from the human mosquito host to human host, both the structures are present. So essentially you can say the XrRNA1 is a conserved element in a hypervariable region in the viral genome. Also, if you do another sequence analysis, it is interesting to show that these pseudoknot structure or essentially the dual pseudoknot structures have evolved only in mosquito-borne flaviviruses and not any other flavivirus. So with that thing in mind, we kept our questions as that what is the significance of this evolutionary conservation of pseudoknot 1 element, and then what is the role of viral encoded miRNAs. So with that, the first experiment which we did is that we infected dengue serotype 2 both in HUH7 cells, the hepatic cells, the site for virus infection, and a mosquito cell line. And here we are seeing that of the 6 potential candidates, 2 of them, that is viral microRNA 5 and 6, were significantly upregulated in both those cell lines. So dengue virus infection leads to significant upregulation of viral microRNA 5 and 6. Now, in order to further show that indeed they are getting produced from a viral RNA and not from any other source, what we did essentially is that we did site-directed mutagenesis such that we have melted these structures. So essentially, if you consider that the viral microRNA production site is in the pseudoknot 1 element or XrRNA1 element, we have deleted both XrRNA1 and XrRNA2. Now for this study, we have used a reporter construct such that when you transfect this reporter construct, the luciferase activity will tell you the effect on the viral translation, whereas the total RNA level will tell you an effect on the replication efficiency of the virus. So if you transfect this construct again, the wild type and mutant construct, in a human cell line and a mosquito cell line, again you are seeing that only upon deletion of pseudoknot 1 structure, which is found to be evolutionary conserved and it is not being mutated across different flaviviruses, you see downregulation of viral microRNA 5. So essentially this shows that indeed this structure which was present and not found to be mutated across different flaviviruses essentially has a significance, that is the production of a microRNA like species. So with that we conclude that XrRNA1 is essential for the production of viral miRNA 5. Now we also check whether our mutants have some effect on the virus life cycle, and we are seeing that there is no effect on the translation of the virus or the replication efficiency of the wild type and mutant viruses. So the wild type and mutant reporter construct do not affect the virus life cycle. Now this was data which put us into that what is the potential role of these microRNAs. Essentially they are not affecting the virus life cycle, their production or something like that. So with that we wanted to check that whether these viral microRNAs can act as host miRNAs. And as we see that in case of a normal host microRNA, it is loaded into the RISC complex, and this RISC complex then binds to a host mRNA and degrades it. So we pull down using AGO2, which is a very important component of a host RISC machinery, and we are seeing a very significant upregulation of viral miRNA 5 and 6 over there. So indeed they have some role by loading and degrading the host mRNAs. So they are loaded in the AGO2 protein. Next, to further show that they have some role in pathogenesis, we check their level in exosomes. So these are extracellular vesicles, they are secreted out upon infection, and they have been shown to play a very prominent role in pathogenesis. So in a cell culture system, when we check the availability of these microRNAs in exosomes upon infection in HUH7 cells, that is human cell line, we are seeing that both viral miRNA 5 and 6 are significantly upregulated in the exosomes, but not in the exosome depleted supernatant. So essentially here what we are showing is that the virus can produce microRNAs, these microRNAs are getting secreted out, and now they can go to different tissues inside the body and cause pathogenesis. So they are secreted via the exosomal pathway. Further, we were parallelly doing another study in which we are checking the level of different host microRNAs in these exosomes, host microRNAs and not viral. Now in this study, we have different patient samples from different plans of severity. That is essentially plan B and C being the more severe form of dengue. Now in those plans, when we check the level of the viral microRNAs, we are seeing a upregulation of a host miRNA MIR96-5P, and also both the viral miRNA 5 and 6 are very highly upregulated in severe dengue patients, which also points out to the possibility of they playing a very important role in pathogenesis. So next, because these are newly identified and novel microRNAs, there are no predicted targets. So we developed a pipeline, and then we predicted a lot of targets which they can bind to and suppress. So in case of viral microRNA 5, we have predicted around 28 nodal RNA targets. That is, one RNA can regulate multiple pathways. That is why they have been termed as nodal RNA targets. And in case of viral miRNA 6, we are seeing around 32 of such targets. Now, interestingly, while doing a lot of analysis in this prediction, we came across a very interesting observation. The MIR96-5P, a host microRNA, which was upregulated in the exosomes in severe dengue, showed targets which were correlating with both viral microRNA 5 and 6. So, essentially, around 12 targets which are involved in viral microRNA 5 functioning are also targets and validated targets of MIR96-5P, and they have been shown to play a role in nervous system. Similarly, viral microRNA 6 targets have been shown to play a role in suppressing the host immune response. And further analysis showed that these microRNAs can actually target either the brain or spleen, which can further cement the hypothesis that they have a very prominent role in these two functions. So essentially what currently we are right now in the stage of working is to validate these targets, both using a mice model and also to show in the specific cell lines that what is happening over there. So with that, this is just a conclusion slide. When the virus infects, it replicates inside the host, it encounters a lot of RBPs. Now this RBP interaction is leading to production of viral microRNA like species, which can target cellular RNAs, and the hijacking of the host secretory machinery by the virus can itself load these microRNAs into extracellular vesicles, and now they are secreted out in these extracellular vesicles, and now the microRNA is not only present at the site where virus is replicating, but rather it can go to the entire body and cause pathogenesis. So with that thing, I'd like to acknowledge ISC and MCB department where I have done my work, and my lab. So this is the current SG lab family, but we have got a lot of seniors who are sitting over here. So with that thing, I'd like to end my talk. So thank you.
Thank you, Ashish. Questions from audience? Brother Das, you want to ask something?
I'm curious about something. I'm just curious about something. So does this essentially imply that there's a lot of redundancy in the whole system? Because you have so many of these microRNAs.
Yeah. So sir, what I can say is that the total secretion of an exosome or microRNA by an exosome is actually redundant, in the sense that only when a virus infection happens or some bacterial infection, the trigger is such that this imbalance, there is an imbalance in this redundancy. So in case of, if I only talk about some host microRNAs, it has been shown that virus infection specifically triggers some host microRNAs which are stable in case of virus infection, and thereby it proceeds to pathogenesis. So if I talk that in case of dengue, it has been shown that there is an overall suppression in the production of microRNAs. But essentially some of those microRNAs are stable. Now we don't know the mechanism, but when those microRNAs are stable, they can suppress the host immune response and thereby trigger the virus infection. So the redundancy is there until it is triggered by some infection.
Even upstream of the production of the viral microRNAs, one can hypothesize that the formation of these double-stranded RNA structures could activate the MDA5 and the mediator data production and the MDA5 signal. So it's probably a way of the virus to mimic and hijack the abortive machinery and then avoid that signal.
Yeah.
So what I'm asking is, have you checked if putting these double-stranded RNA structures activates the MDA5 pathway, and whether abortive inhibition is viable antiviral approach?
So essentially, it has been shown that dengue virus or any other flavivirus give rise to two RNA species. One of them is small flaviviral RNAs. So these are long stretch, around 200, 300 nucleotides RNAs, which are produced from the 3' UTR of the virus. So they themselves can act as double-stranded and then trigger the MDM2 pathway, which you are saying. But in case of viral microRNAs, what our preliminary results suggest is that anyways, in case of infection, the MDM2 pathway is triggered. But it has not still been shown that how dengue virus can cause pathogenesis in the brain. For that example, there are reports that the exosomes can cross the blood-brain barrier and then enter the brain. So our preliminary targets, what we have identified, essentially is pointing out that though it is an accessory mechanism for the virus to produce such, or they might be getting produced as a result of like the mechanism which you just pointed out, but indeed they have a role, and that is what we are currently working. Yeah. Yes.
The dengue virus 5' and 3' UTRs are involved in circularization of the genome, right? So when you delete these motifs in the 3' UTR, are those motifs affected? Is the circularization of the viral RNA affected?
Yeah. So the deletions which I did are based on a literature in which they have shown that essentially the overall circularization is not affected. But since I am deleting the top 4 nucleotides of a pseudoknot, that pseudoknot is not getting formed. And that pseudoknot, essentially the pseudoknot 1 element, has not been shown to as such play a role in circularization and life cycle of the virus. So essentially its role has not been discovered yet, but this is what we think, that is the presence of that RNA structure is essentially for this role. But yeah.
Have you looked at subgenomic RNA fragments, whether their formation is affected?
Yeah, so if you delete pseudoknot 1 element, essentially the subgenomic RNAs which are getting produced are produced from pseudoknot 1, pseudoknot 2, DB1 and the last stem loop structure. So if you delete one of them, the idea is that XRN1 has now a free site. It can just bypass pseudoknot 1, go to pseudoknot 2. So essentially what we have seen is that if you delete pseudoknot 1, there is a more production of viral microRNA 6, which is a downstream of viral microRNA 5. So indeed it is producing more subgenomic RNA, but the part which we are pointing out that these subgenomic RNA are acting as a precursor for the production of viral microRNAs. You can correlate as them that subgenomic RNAs are actually pre-microRNAs if you compare with host microRNAs, and viral microRNAs are the mature microRNAs. Thank you.
Ashish, last question. This packaging or co-opting of exosome machinery and packaging of microRNAs, it's happening on purpose or it's happening on its own randomly? Should not happen randomly, there must be some purpose.
Yes, sir.
There's energy being used to make them.
Yeah. So it is purposeful in the sense that the exosome machinery as such can take both proteins and microRNA. So the effect of different proteins which are secreted out by this exosome machinery has been shown. Indeed, the dengue virus NS1 protein itself can be loaded into the exosome, and it is known to be a very pathogenic protein. So what I like to say is that essentially it is a very well regulated process, and somehow the virus is hijacking it. Now when the virus hijacks, it takes over whatever is going inside. So it might be binding to some proteins and these microRNAs are getting along, or some proteins are coming out from the cytoplasm, but everything is regulated by the virus. So that is what's.
Thank you so much.
Thank you, sir.
So, round of applause for all the short talk speakers. And I'll stop talking here because students are waiting eagerly for announcement of the awards. I'll give it to the session actor, Rajesh now. Please take
Okay. So the most awaited session for the last 2 days, the prize distribution session. So first we'll start with the short talk speakers. For which I would request Professor Samitra Das to please come on stage. The first speaker, not by the order of their talk, so it's Amrita Ray from Soma Chattopadhyay's lab from Institute of Life Science, Bhubaneswar. Please put up a round of applause. The next speaker is Ashish from Samitra Das Lab from Department of Molecular Biology and Cell Biology in IISc. The next speaker is Ghulam Hussain Syed from Institute of Life Science, Bhubaneswar. The next speaker is Deepa Singh from Krishna Inampodi Lab from AIMS Bhubaneswar. Sorry, AIMS New Delhi.
Very nice talk and excellent work.
The next speaker is Abhik Shah from Presidency University, Kolkata.
Great.
As Vinay sir had a flight to catch, so he left early. So next, move on to the next session. No.
Oh, the poster award.
Yeah.
Oh, okay.
So for poster awardees are not any specific order. It's P. Sai Pujita from CCMB from Krishnan Harshan's lab. These are the top 5 poster awardees. It was very difficult because the top 70 were selected very difficultly, and after that selecting the top 5 was very, very difficult for the judges. The next awardee is Onam Singh from Thisti from Milan Sujit Lab. The next awardee is Buddhadev Baral from IIT Indore Hemchandra Jas Lab. The next awardee is Gaganpreet Kaur from Aiser Mohali from Indranil Banerjee's lab. The last awardee is Suraj Jagtap from Chemical Engineering Department from Raul Roy's lab. Any other lab member or Raul Roy's here? I'm not sure.
Congratulations to your lab and the group.
Thank you, Samita Das sir. Now let's move on to the next session where I would request Dr. Keshav for the giving a vote of thanks for the 7th Virology meeting.
first of all, I thank all of you for attending this meeting. And I would like to ask you guys to give a big round of applause for all the speakers, panelists, and the chairs and attendees of the TISTA. Please go ahead. And also I would like to commend the efforts of all the volunteers from our labs, from Professor Samitra lab, Shashank lab, and my lab. They're tirelessly working for about a week, and that's why we could able to sit for some talks in this meeting. And give them a big round of applause for all the volunteers. Now, just to conclude the meeting, and you must have heard about the talks, and it was fantastic talks, especially with respect to the fundamental virology, how some people are taking them to the clinic, and also to the translation. We could hear that some of the mRNA vaccines are already in the clinic, which is commendable for us, because we can see that from fundamental science to the translation, we could be able to do it at Indian level as well. And at the same time, I'd like to thank Professor Samitra, because he was the one who has been visioning this for some time, and me and Shashank would be able to get that onto the platform with the help of him for having all of you for this meeting. And I thank Professor Samitra, and I really enjoyed working with him and Shashank. And thank you all, and I really hope you like the meeting and you enjoyed the stay here. And I would like to thank Sri Kumar for accepting the next meeting, because now we know that where is going to happen, and we all look forward to having you in Kerala soon. Thank you all, and let's go for the high tier right now.
Just a small session. With the hope that we all will meet next year in Trivandrum, I would like to ask Professor Soumitra Das to give a concluding remark. From there, sir, can you use a mic? Yeah.
So essentially we have already thanked, but I think on behalf of all the audience, we would like to thank the conveners, Dr. Shashank Tripathi and Dr. Keshav Kanula. They did tirelessly job, and as I said in the beginning, that they are showing that the young blood can actually take it forward. See, we have shown we have initiated something, but these things will be continued, and all of you will be in future great virologists, and this virology group will contribute to your endeavor. All the best. Good luck.
Please move for the high tea. Thank you. So I leave the rest here.